Sodium dodecyl sulfate polyacrylamide gel electrophoresis (Sds-Page) is a method that separates proteins based on their molecular weights. Sds-Page is a discontinuous electrophoretic system developed by Ulrich Lemny. It is commonly used as a method to separate proteins with molecular masses between 5 to 250 kilodaltons. It is a technique widely used in biochemistry, forensics, genetics, and molecular biology. SDS is a detergent, an anionic surfactant, so in other words, it is a negatively charged compound that lowers surface tension. In the case of proteins, SDS disrupts the non-covalent bonds in protein molecules. Page is a biochemical technique that allows for proteins to be separated by their electrophoretic mobility, meaning that how fast they move in an electric field. In the case of Sds-Page, they are separated by their size, but not according to their charge. In principle, SDS detergent denatures proteins by binding to the hydrophobic regions. Upon this binding, all non-covalent bonds get disrupted and proteins acquire a negative net charge. A concurrent treatment with a disulfide reducing agent such as betamersopteethanol or DTT further denatures the proteins by reducing disulfide linkages, thus overcoming some forms of tertiary protein folding and breaking up quaternary protein structure. So the protein samples are having uniform structure and charge, and therefore the separation will depend on their molecular weight only. The number of SDS molecules that bind is proportional to the size of the protein. Thereby, in an electrical field, protein molecules move toward the anode and separated only according to their molecular weight. Small proteins migrate faster through the gel under the influence of the applied electric. Here you can see the list of apparatus that we need. We need DC power source, reservoir or tank, glass plates, spacers and cones, sport medium, polyacrylamide gel, buffer system with high buffer capacity, molecules to be separated such as proteins or nucleic acids. Here is a methodology for Sds-Page. Firstly, we need to prepare a polyacrylamide gel, and then we need to prepare the samples. Then, for the denaturation of protein, samples must be heated to 95 degrees Celsius for 5 minutes. Then, we load the samples onto the gel. Next, we start running the gel at 200 volts for 30 to 40 minutes. Finally, we need to stain our gel. First, we need to prepare our sample. A typical protein load for a sample of protein is between 5 to 20 micrograms per lane. Too much protein will distort the bands, and too little protein load will be difficult to detect by coma staining. Loading dye is used for the preparation and loading of protein samples onto a gel for Sds-Page analysis. SDS contained in the sample buffer is used to denature proteins and make them negatively charged. In this manner, each protein will migrate in the electrophoretic field in a measure proportional to its length. The gels often used in SDS are considered discontinuous gels. These are gels that contain both a stacking gel and a separating gel. When glycine from the upper reservoir enters the low pH of the stacking gel, it will principally be in the chloride ions now carry the current and migrate toward the anode. During this step, chloride ion concentration becomes lower at the top of the gel and higher at the bottom of the stacking gel. As electrophoresis continues, protein molecules, which are negatively charged due to SDS, will become greatly retarded, allowing the trailing protein molecules to catch up. The stacking gel is very low in percent to ensure there is little separation based on the pore size. This will ensure all of the proteins enter the resolving gel at the same time to get small tight bands. Then we can start running the gel. Running buffer in gel electrophoresis are used to provide ions that carry a current and to maintain the pH at a relatively constant value. These buffers have plenty of ions, which is necessary to allow the passage of electricity through. After running a 12% Sds-Page gel, the gel is stained with Comessi blue. Comessi blue is a dye that will bind to the acidic amino acids in proteins. This method of staining will stain most proteins in the gel with as little as 0.1 microgram of protein in a single band. Washing off the gel with this staining solution will remove the unbound dye from the gel, but not the protein, leaving behind several nice blue bands that show each protein. Here you can see the apparatus for casting the gel. There are 4 apparatus for gel preparation. Here we have two glass plates. One is shorter, the other one is taller. We place the short glass in front of the tall glass and put it into the casting frame. We have also a casting frame. After placing into the casting frame, we secure the plates and check whether the bottom is flat. After these steps, now we can place this onto the casting stand. Firstly, we prepare 12% separating gel. Here you can see the recipe for this gel. We'll prepare the mix in this order, starting from 1.5 molar tricyclic chloride, acrylamide stock, water, 10% SDS, 10% ammonium persulfide, and TEMET. Then, we pour this mixture in between the glass plates. After preparing the gel mixture, we leave it to polymerize at room temperature for 30 minutes. After 30 minutes, we now need to prepare stacking gel. Here you can see the recipe for stacking gel. We again follow the same order. This time, tris chloride has a pH of 6.8. Then, we pour this mixture in between the glass plates. Then we place the comb. Then we wait for the gel to polymerize. After gel is solidified, we place it into the running compartment. Because we have only one gel, we need to balance it with another plastic one. Comb must face towards the inside of the gel compartment. After we pour the running buffer, we need to make sure that there is not any leakage. Then we can start loading the protein samples. We should now remove the comb gently. Firstly, we need to load the protein marker. Then we can start loading our samples. Then refill with running buffer. The gel box has two electrodes with black and red color coating. The inner gel compartment also has these matching colors in black and red. These colors must be aligned, as red on red and black on black. We adjust the settings to 80 mA and 120 volts. We then let the gel run until the dye is just at the bottom. It should take around an hour. We should not stop the gel too early. The results will be difficult to obtain because the proteins will be fast resolved when the gel has run all the way. Bubbles coming out is an indication of the system is running. After the run, we turn off the electric supply. We then take the gel cascade out. The running buffer can be used multiple times, so we pour the excess buffer back into the gel box. Take the gel out of the apparatus. Remove the short glass. Then slice out the part of the gel with wells. Now, we will stain the gel for visualization. We will add staining solution and keep the gel on shaking incubator for 15 minutes. After 15 minutes, we will transform the gel to another box with destaining solution. The gel is then put back on the shaking incubator and incubated overnight. Here is the gel result. At the very left, we have a protein ladder. Let's imagine this gel has bacterial lysates with different proteins. As you can remember from bacterial protein expression and purification video, we can induce a particular protein's expression by IPTG. In this first lane, we have IPTG-induced bacterial protein lysate. We can see that there is an overexpression of that particular protein at 25 kilodaltons, along with some other proteins at different sizes. In the second lane, we have a negative control, in which no IPTG induction took place. Therefore, we can see the overall protein content of the lysate, however, there is no intense band around 25 kilodaltons. In the third lane, we have a purified sample after affinity chromatography. As you can see, the most intense band is at 25 kilodaltons, so we can say that this protein is purified relatively well. In this video, we showed how to set up a Sds-Page experiment and separate proteins by their molecular weights.